All right, you guys, picking up where we left off, we finished chapter 20, our version. Now we're going to pick up chapter 21. Okay, so that starts on page 781 if you're following along with your text. So let's break down the immune system, which takes place all over inside of the lymphatic system, which it's not really an organ system because while it has vessels and organs, it also just has this diffuse tissue associated with it throughout the body. But again, the lymphatic system is kind of like the brick and mortar where all this stuff happens. And now we're going to talk about what actually happens, okay? And we break down what actually happens in two main levels to begin with. You have your innate defenses, which are the ones that you're born with, simply speaking. These include your surface barriers like your skin and mucous membranes, right? Because remember, you are a donut. So your epithelium is your skin on the outside of your body, but it continues into and throughout your gastrointestinal tract, right? Your digestive system. All of that is considered to be exposed to the outside world. So your surface, your body's surface includes both your keratinized epithelium and your mucous membranes, which is basically just your non-keratinized epithelium of your food tube, right? It also includes other general internal defenses, which include biological defenses, and all the cells associated with that. So you have phagocytes, which go around eating bad things, generally general bad things. You have natural killer cells, which go around killing general bad things. Inflammation, a response that your body has to general bad things. Antimicrobial proteins, they have the purpose of inhibiting infection and basically like getting in the way of bad things. And fever, which is a really important response that your body has to really any living pathogen. Raising the temperature helps. So your innate defenses that you're born with include both these surface barriers and your a group of different cells and or proteins that basically attack anything generally. So they don't have to be taught. They just recognize non-self and go for it. The other half of your immune system, and the one that you really think of when you think of your immune system, is called your adaptive or your acquired immune system. Okay, so it's also known as your, or you may see referred to as your acquired immune system, as in you acquire it later on in life, okay? This is the one that needs to be taught. It's the one that makes your antibodies that learn to recognize a specific antigen associated with a particular pathogen, what have you, and then basically remember that for next time, for the potential next time. That adaptive part of your immune system can be further broken down into humoral immunity, which is immunity, immune system functions that occur within the humor, within the fluids of your body, your interstitial fluids, your extracellular fluids, the stuff that's not within cells. The other half of the adaptive or acquired defense part of your immune system is the cellular immunity, which happens within cells, okay? So one happens outside of cells in the humor, we call it humoral, and the other is happening in cells, we call it cellular. Your B lymphocytes that we talked about in our last lecture, last half of the lecture in the lymphatic system, those are they're prominent in your humoral immunity, while the T lymphocytes are more prominent in the cellular immunity, okay? So we're gonna start off by talking about innate defenses. So I'll have a header saying that this is innate, 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 and then at some point down the road, we will switch over and start talking about these adaptive defenses, and this is where it's really gonna start to get kind of hairy over in here, okay? So bear with me. Let's talk about those surface barriers for starters, okay? So innate defenses, surface barriers. So again, innate defenses is the stuff that everybody has. It's innate in you, okay? You have skin, right? Your intact skin epidermis. The physical barrier of that. It's literally waterproof, right? It's a physical barrier to keep physical stuff out. A mechanical barrier prevents entry of pathogens and other harmful substances. In addition to that, it's also a chemical or has a chemical barrier built in. So the acid mantle of the skin is the chemical environment produced by the bacteria that live on your skin, and like things like sweat, stuff that is constantly on the surface of healthy skin that creates an unpleasant or unlivable environment for bad pathogens, for bad bacteria, fungi, what have you. The keratin itself in the keratinized part of your epithelium is going to give you resistance against toxic substances. Okay, so the keratin, it provides a physical barrier for like microscopic pathogen kind of stuff, but they're so small, like they could go in between cells, but large molecules, acids, bases, bacterial enzymes, it even keeps those out as well, okay? All right, because it's epithelium, remember it has those tight junctions. So the cells of your skin are very, very tightly close together. So they can't go, so nothing can really get through unless there's a break in between them, okay? Or if something bad, a toxic substance, enzyme is produced by bacteria trying to digest away the keratin of your skin, that's the only way that it can really get through. Injury or like a burn of like a chemical burn or something. Anyway, that's the keratinized epidermis. Your food tube is your unkeratinized epidermis, epithelial tissue, I should say. And it includes a built-in mucous membrane all throughout, right? So that mucus, those mucus forming cells that are part of the epithelial tissue of your gastrointestinal tract, including your mouth and esophagus, that mucus is physically going to catch debris and even pathogens before they can get very far. So it is a mechanical barrier, the mucous membrane itself. The mucus itself traps microorganisms in your respiratory and digestive tract. So this includes also like your entire pharynx, so the back of your nose and throat, your trachea down into your bronchioles, mucous membranes, catching stuff and sweeping it out. Your esophagus, your stomach, your intestines, all the way down the line, mucous membranes to catch stuff and sweep it out. Your nasal hairs, so further on, in addition to the mucus in your nasal cavities, you also have hairs that also help to trap stuff and allow it to kind of stop before it gets into your lower respiratory system, and basically gives the mucus a chance to catch it and sweep it out. Cilia is the sweeping it out part. The cells, the epithelial cells of these mucous membranes are ciliated epithelial cells, and the cilia will beat and basically push the mucus and whatever is trapped in it up and out of the body. Hence, snot and stuff like that, right? That's all your body getting rid of actual contaminants. Gastric juice. If something were to get past your mouth or your nose and you swallowed it and got down the esophagus, if it gets down into the stomach, it's done because of stomach acid. Stomach acid will kill pretty much all that stuff. Anything that you cough up out of your bronchioles, out of your trachea that gets into the larynx area, when you cough that up and swallow it, you really kind of should swallow it. That's totally normal because if you swallow it, it's going down into the stomach and the stomach acid will kill whatever is in there that you coughed up. So don't spit. It's not sanitary for everybody else. And it's also like kind of goes against our design. You can swallow that stuff. It's fine. Your acid mantle of the vagina. Okay, so if you have a vagina, Then you have a mucous membrane there that also has its own acid mantle. The vagina is an acidic environment. It's designed to be so to prevent colonization from bacteria or things like yeast, right? It's a self-cleaning system and it's acidic, so it's supposed to take care of itself, and it will if you let it. So if you leave it alone and don't put a bunch of weird stuff in there, then it will take care of itself because of this. Lacrimal secretions, your tears, right? So remember the lacrimal duct is up here and the tears go down over the surface of your eyeball. They coat the surface of your eye. They actually have antimicrobial properties, those tears do. So as they're going across your eye, they're not only lubricating the eye, they're not only carrying with it, whatever stress hormones and energy that's coming from the emotion that caused you to produce the lacrimal fluid, tears, but it also is cleaning the eye. Okay, very important that it cleans the eyes. The substances in it are actually proteins, are actually enzymes that like digest bacteria and microorganisms. And your urine is normally slightly acidic, which is really important too, because every time you urinate, you are flushing out the urethra, right? So your urethra is another opening in your body that stuff could potentially get up into. So having acidic urine flushes those things out of the body if they try and crawl up there. Microscopic things, right? All right, stuff that is susceptible to chemical defenses. All right, so those are the exterior physical defenses. Keratinized epithelium, mucous membranes of the epithelium of your digestive tract, your respiratory tract, your reproductive orifices, right? And your urinary orifices. All of that is very important. It's all very innate. It's all stuff that you were born with most of the time, right? Internal defenses of the innate part of the immune system, okay? Still stuff that we are all born with. We all have this. We have what are called phagocytes or phagocytes. These guys are large cells that specialize in phagocytosis, which is cell eating. We learned about phagocytosis, endocytosis, and exocytosis back in part A. So hopefully you recall that cells that phagocytize other cells have this ability to create these outcroppings of their cytoplasm called pseudopods, and they basically reach out like arms and wrap around a bacteria or a virus or a bad protein or just debris, whatever, wrap around and basically engulf it. When these two parts meet, they fuse and you have a lysosome, a vesicle inside of the cell. You merge that with something with enzymes in it, like a lysosome. Then you can digest the contents, whatever that bad thing was, and exocytize anything that you don't need. You can break it down into small molecules and recycle some of it, but some of it is just waste. And this, remember, this waste, all of this is going out into the extracellular fluid, right? It's going out into the ECF, which means that it's going to go up, it's going to be uptaken, it's going to be taken up by the lymphatic system, where it's going to get cleaned up and everything, all the good stuff is going to get returned to the bloodstream at the ducts, at those ducts, right? Up here, the thoracic ducts. All right. Or subclavicular, whatever. All right. I don't need you to know the names of specific ducts, just that there are ducts, and that's what they do. They go from the lymphatic system into the cardiovascular system, your blood vessels. All right. Okay, so that's how phagocytosis works. The two major phagocytes of your innate immune system are called neutrophils which are really common. They're throughout. They're just basically hanging out in your bloodstream at any given time. You take any blood sample, most images of a blood slide, you're going to be able to find neutrophils. They're everywhere. They're very abundant. They're just, they're immune cells. They go around eating bad things generally, right? Because this is innate kind of general defense. And then macrophages are these relatively very large cells that just go around consuming everything. They're very voracious. They eat a lot. Very hungry. Hungry, hungry hippos. Right here, right? Okay, so you have your phagocytes, which do phagocytosis, eating stuff as they find it. You also have these natural killer cells, right? These guys are these amazing cells, part of the innate immune system, so everybody has them for the most part. These guys are also floating throughout the body, in the bloodstream, in the extracellular fluid, wherever. They're just kind of like patrolling around the entire body at any given time, and they attack anything that don't have self-surface antigens. So basically, every cell in your body has proteins on its surface. Remember, we talked about this in part A too. These surface proteins that are embedded in the plasma membrane that basically are identifiers. They're basically telling the immune system, like, hey, I'm supposed to be here. It's their pass, right? It's their multi-pass. You have to have this before you can go, or else you get eaten. If you do not have MHCs, which stands for major histocompatibility complex, if you don't have a major histocompatibility complex, like you should as any human body cell of you specifically, these natural killer cells, they will take care of it. How they do this is not through phagocytosis. They don't eat it like a phagocyte does. Instead, they call over complements, what are called complements, complement proteins. So they call over these specialized proteins whose job it is to, when they are alerted to and directed to a target cell, they will create a little complex in the plasma membrane. They insert themselves in the plasma membrane. They literally form a hole in the plasma membrane of the offending cell, the targeted offensive cell, which allows water to flood in. And we remember when we talked about osmosis, that water will come in down its concentration gradient, right? So bacteria, something like this bacteria, are constantly pumping water out because the inside of the cell is more concentrated with solutes than the outside, therefore water keeps trying to get in. If we allow the water to flood in, then the cell will eventually burst, and we call that lysis, right? It'll lyse. The cell will lyse. And that's a lysed bacterium right there. The natural killer cells also aid in inflammation response. So when they encounter a situation like a cut or something, bad things in the body, they will also send out signals to the rest of the immune system. And that includes parts that will promote an inflammation response, which we're going to talk about here. Okay. So the inflammation response, this example is in response to an injury. You stepped on a nail, okay? So when you step on a nail, you've got, this is probably bacteria or whatever gross crap is on the surface of the nail that ends up inside of your body, past the epidermis into the dermis, right? Looks like here, which means that it will bleed. Remember your integumentary system. It has to pass the epidermis and go into the dermis before we get any blood vessels here. So if we have gone into the dermis, then we may have damaged some blood vessels. Those blood vessels then will, of course, be open and you will have bleeding. All of these cells will be going into the site one way or another, right? In addition to that, however, your whatever cells happen to be in the area, like your natural killer cells, for instance, part of the response that they're going to trigger with literally just chemical signaling is going to be vasodilation. So the blood vessels that are around that, whether they're broken or not, basically they will widen to allow more blood to come into the area. The epithelial cells that form the wall of these blood vessels will actually separate from each other a little bit, like they'll actually leave a gap. Then that means that the leukocytes, the white blood cells that we talked about, well, we didn't really talk about leukocytes yet, but they're immune cells that are flowing through the bloodstream. They are going to end up being able to squeeze through those little gaps in the wall of the blood vessel. They will reach the site of the injury, and then you're going to, then they're going to basically do their thing. So the inflammation response is uncomfortable. We associate it with redness, swelling, pain, and heat, which are all actually very important parts of the inflammation response. But the importance of it is that it prevents the spread of the whatever damaging factors that have been introduced, prevents them from spreading to other areas. It's going to dispose of debris, whatever has gotten in here. Maybe not the nail, that might be up to you. You'll probably have to remove the nail, but any little like bits of rust or horrible things that are on there, dirt, and whatever pathogens are in there as well, that all has to be cleaned up by your macrophages, your neutrophils. These guys are going to come in and phagocytize all of that stuff, right? And sets the stage for repair. Let's take a look at this flow chart because I love flow charts. This one's out of your text, page 787. At the point of tissue injury, you've got two things happening. You have inflammatory chemicals being released. Basically, that's the signal to the rest of the immune system that, hey, come over here, something's up, right? It includes histamines, complement kinines, and prostaglandins, among others. The only one of these that I really am interested in you knowing are your histamines and your complement proteins, okay? So your histamines are, they're part of the inflammation response. They're the ones that we associate with the inflammation response. You take antihistamines like Benadryl, stuff like that to try and tamp down this inflammation response if it's like for an allergy or something, right? So your histamines are basically molecules or chemicals that help with the inflammation response, while your complement proteins like I said before, they get called in to actually lyse pathogens. So they get called in to come in. They are pointed to a particular cell, like a bacterial cell, like this guy, make a hole and cause it to burst, right? That's the complement proteins. That's what they do. So as a result of these chemicals and proteins, these molecules, you're going to have the arterioles dilate. So the blood vessels are going to dilate, get wider, greater blood flow. You're going to have the capillaries become more permeable. You're going to have the epithelial cells kind of separating, causing gaps so that stuff or immune cells can come through the blood vessels into the interstitial fluid. And the chemicals will also attract other immune cells like your neutrophils, your monocytes, lymphocytes. We call this process chemotaxis. This is, you can just refer to this literally as attracting immune cells more generally. It's basically calling for backup, okay? Let's see. This one, I'm not, this This portion of it, I'm not as worried about you knowing. The leukocytosis, basically leukocytes, they're basically just called to the area to do phagocytosis, to clear the area. You will also have other phagocytes being called in. Phagocytes clear the areas, basically what that means. While your capillaries are leaking fluid, allowing cells to come through, clotting proteins to help stop bleeding after everything has arrived. After everybody's arrived, you can stop the bleeding, right? Increase blood flow to the area. Here's your 4 factors that we associate with inflammation. That is what inflammation is. We form a scaffold, and that is basically laying the stage for healing. So from then on, you're basically rebuilding the damaged tissue. Okay, so tissue injury results in the release of chemicals and proteins that call forth other immune cells, important immune cells, including phagocytes of various kinds that clear the area of debris, including pathogens. You have the chemicals are going to allow your vessels to dilate to increase more blood flow so that these guys can get to you faster. It's going to cause the capillaries to leak fluid, allowing more of these cells to get into the area faster. The combined result of these things is that you're going to have swelling of the area. The swelling is going to probably press on nearby sensory nerves, so therefore you have pain. You're going to have redness from all of this blood flow, and you will have heat from blood and fluids traveling from deeper in the body to this area. All of those things are there for an important reason, okay? All right, more of your innate defenses. Aside from inflammation, which is very generalized, right, occurs with any injury, you also have what are called antimicrobial proteins, specifically interferons, okay? So not only do you have cells like macrophages and other phagocytes that are there to, or natural killer cells that are there to kill pathogens, right? You also have proteins who can impede pathogens, or kind of like a citizen's arrest. They're going to kind of like hold them in place or bother them to the point where they cannot do harm to other cells. So if a cell is infected by a virus, right, what a virus does is it injects its RNA into, or DNA into one of your body cells. That DNA goes into the nucleus and basically inserts itself into your DNA. And it's basically just instructions to make more virus. So your body goes about its business of replicating its DNA and translating it into proteins. And before you know it, that cell has basically been hijacked and turned into a virus factory without knowing it. It knows enough, however, when this has happened, to also translate genes for these proteins called interferons. So if a cell gets infected, it will also create interferon proteins, which it will release so that they can get to the surrounding cells. It's basically like a save yourself signal, okay? So interferons are secreted by the infected cell, which basically has no hope, but they can reach the surrounding cells in time for the surrounding cells to create antiviral proteins that can stop the virus before it gets into that cell. Okay, so interferons interfere with the way that the virus behaves by basically warning surrounding cells, and the surrounding cells can then create antiviral proteins to stop the virus from getting in, which is pretty awesome. You don't need to worry about the different types. Interestingly, they may have a cancer-reducing role, which is really cool. So a cancerous cell knows enough to know that that something is wrong, basically. So it may secrete interferons to surrounding cells to warn them, or to tell surrounding immune cells like, hey, I've got a real big problem over here. You might want to come and take care of it, right? Okay. So interferons, kind of like the save yourself proteins. Okay. All right. You also have, in addition to interferons, other antimicrobial proteins called the complement system, which I've already mentioned. So here's just a kind of a closer look at what these complement proteins do. They are basically called to the offending cell, the pathogen, in this case, maybe a bacteria, again. They are going to, they could be called by a lot of different immune cells. So that's why they call it the complement. So you have immune cells that actually kill the bad guy directly, and then you have immune cells that call complement proteins to get it, right? And so the complement proteins complement these guys by helping them do their job. So once they are called in, we call it, well, the classical pathway, there are 3 ways that this can happen. The classical pathway is that they are activated by antibodies coding the target cell. So they may not actually come into contact with the immune cell, but antibodies are basically like tags on the bacteria saying, kill this one. Okay, so antibodies are like markers or tags. You've been marked for death. So therefore, these complement proteins, when they find you, are going to make a hole in you and cause you to lyse. The 2 other pathways, not super worried about you knowing. Lectins are basically just a different type of marker that the complement proteins can recognize as a marker that that cell is marked for death. And if the alternative pathway is that literally some complement proteins or immune cells that call complement proteins recognize a cell that doesn't have those MHCs, right? That major histocompatibility complex. If you don't have that identifying marker, that may be enough to bring in the complement proteins to come and put a hole in you and cause you to lyse, okay? All right then. Okay, so to wrap up, or to not to wrap up, to summarize, to wrap up this part, to summarize the innate defenses, okay, the stuff that everybody's born with. The internal defenses of the innate defenses. So innate defenses has both an external component, your skin and mucous membranes. We're talking about the internal component, all the different cells and molecules that we have that can do a general good job of killing invaders by causing inflammation or just by roving around killing anything that looks bad. Here are all of your internal innate defenses. You have your phagocytes, which go around eating things. You have your natural killer cells, which go around not by phagocytizing, but they go around killing things by calling over complement proteins, possibly, or basically just initializing apoptosis in the bad cell. The inflammatory response, which involves a lot of different cells and molecules. An injury happens, or something is recognized as being an antigen or non-self, and you call in the troops, basically, leading to heat, swelling, pain, and redness. You have your antimicrobial proteins, your interferons, which are the save yourself signal, right? That an infected cell might secrete to save the surrounding cells, or to help the surrounding cells to prepare to fight against the virus that they're infected with. Complement proteins are the ones that they complement the rest of the immune system by basically coming in and doing the dirty work for other immune cells. They're the ones that actually insert themselves in the plasma membrane and form a hole which causes the water to rush in and the cell will burst, or lyse. The last one is fever, which is really interesting. So some of your immune cells, this is systemic, right? So this is not localized to the area of the cut. So it's a different thing than the heat associated with like a cut or an injury, the inflammation response that you would see there. Fever is systemic. So it's over your entire body, throughout your entire body. Basically, immune cells are going to go up and change the thermostat in your hypothalamus, which is really crazy. So they go up, they turn up the thermostat, and it turns out that that heat might denature lesser microbes, if you will, faster than it denatures like your proteins that you actually need. So it can help to fight things like that. But it also increases your metabolism, which increases the speed of your immune system. You are going to inhibit microbes from multiplying with a higher temperature. And then that metabolism is also going to speed up any body repair processes that are happening too. So fever is going to speed up metabolism, speeds up the immune system. It causes the liver to sequester certain important nutrients that the microbes might actually need. So if the bad bacteria uses certain factor, chemical, molecule, the liver will actually sequester that and keep it away from them. And the heat itself will slow down pathogens, keep them, it makes a harsh environment for them to reproduce in, and it may actually denature their proteins, which is usually their outer shell, their plasma membrane made of proteins, killing them outright just like that. So different types of cells, different types of proteins, and then fever, the systemic response of fever, and the localized response of inflammation, right? Cells, local, fever is systemic, and these guys are proteins. So these are all of your internal, your internal innate defenses. Okay. Remember, your external is your skin and your mucous membranes. Internal. Okay. All right, so that concludes the innate defenses. Okay, so that's the stuff that everybody's born with. We also have the adaptive or the acquired defenses. And these are the ones that you think of when you think about your immune system. It's the part that actually learns about an antigen and produces antibodies for the next time they encounter that antigen. It's what provides you with what we call immunity, okay? It's what vaccines help us to do, right? It's why when you catch chickenpox once when you're a kid, you don't tend to get it again later on. Your body remembers it and takes care of it much faster the next time that it sees it. So we've got those B and T lymphocytes, okay? These guys are the major key players of your adaptive defenses, the adaptive part of your immune system, right? Your B and T cells. Your adaptive defenses, your adaptive immune system is very, very specific. So the T cells or the B cells, the memory that you have, the antibodies that you make are very, very, they're literally specific to one antigen, and that's it. They recognize and respond to specific diseases, each one of them, okay? There are more than one for each disease, but each one of them is only programmed to recognize one disease. It is systemic. Your adaptive defenses are throughout your entire body. Your innate defenses, like the inflammation response in response to an injury, that's localized, right? That's just in one little area. Everything's drawn to it. But your adaptive response, if a lymphocyte, B and T, are activated by an antigen, they are going to spread throughout the entire body, hunting that down. All right, in one way or another. And it has memory, of course, right? Your immunity, the antibodies that you get from this adaptive immune system is like the part that we think about when we think about the immune system. After your initial exposure, it's able to recognize that pathogen and initiate a stronger response the next time. Stronger and faster. Okay, the functions of your adaptive immune system is it has the ability to stimulate specific lymphocytes to proliferate. Okay, so you have what's called immunogenicity. So basically, that again is that specialization. Okay. For a particular disease. Reactivity, you're going to have a reaction. Again, this is a systemic reaction throughout the entire body. The activated lymphocytes are going to create antibodies as a result of the first contact, okay? Your antibodies or the lymphocytes, they are going to, the receptors of those lymphocytes, so your B cells and your T cells, just like any other cells, they have receptors on their surface, but these of course are going to be specialized for a particular disease. The way that they recognize the disease is by a specific antibody, okay, which are produced by those B and T lymphocytes, okay? So antigenic determinants are going to be the receptors that particular antibody has, which is able to attach to one specific antigen. Okay, so this is again kind of the specialization. So like this antibody can only talk to an antigen with this antigenic determinant. An antibody with a different type of receptor can only see or respond or bind to antigens with this antigenic determinant, et cetera, okay? And another important function, a very, very important function of your adaptive response is those major histocompatibility complexes. This is critical. Your cells, your immune system needs to be able to recognize your cells as opposed to other cells, right? So self versus non-self. So any kind of a pathogen is considered non-self. Any kind of a molecule that's not recognized as being produced by a human cell, technically non-self. A blood transfusion of a different blood type, non-self. Transfusion or transplant of an organ from a donor, it's a big problem, right? Your body looks for these MHC proteins on the surface of every cell that they encounter to make sure that it is self. And if it's not, then it will destroy it. It will try to destroy it. Okay. Okay, more functions. So more specifically now, we talked about lymphocytes generally, B and T cells. Let's talk about them a little bit more specifically. Your B lymphocytes, again, are going to be involved in your humoral immunity. Again, that is the immunity outside of the cells and the extracellular fluid, right? The T lymphocytes are involved in your cellular immunity. Which does not involve creating antibodies, okay? The B cells are where it's at when we talk about antibodies, really, okay? They're the ones that are making the antibodies. APCs are antigen-presenting cells. So T lymphocytes, they don't create antibodies, and they don't often, or they don't always attack cells directly. Sometimes they need to be educated about what to attack. And how that happens is that antigen-presenting cells, these specialized cells. I'm gonna actually change this to the cells of the adaptive defenses. These are like the key players. These guys are going to actually take a chunk of the antigen that they probably have just killed and put it on their surface. When the APC encounters a T cell, that T cell will recognize the antigen because it is coming from an APC. If it finds the antigen on its own, it won't recognize it. This is a really important safety check for your immune system. If T lymphocytes, if T cells were allowed to decide for themselves every single time what to and not to kill, you probably, well, you'll be more likely to end up with an autoimmune problem where you're going to be attacking your own body cells. Having APCs as like a middleman to introduce an antigen to a T lymphocyte so that the T lymphocyte needs to have this middleman before it can attack anything or recognize something as an antigen. It's kind of just like a secondary layer of protection for your own system, for your own body against your own immune system. Okay, so APCs, antigen-presenting cells. They present the antigen to T cells, okay? All right, this is tough to read, I apologize. So again, your B cells and your T cells, they both originate in the red bone marrow, remember, okay? So you've got your little non-differentiated lymphocyte precursors, how cute. Some of them stay and become B cells. They mature in the bone marrow, become B cells. The other ones leave and go to mature in the thymus, okay? To become T cells, okay? So some of them stay and mature into B cells in the red bone marrow. Others leave and mature into T cells in the thymus. That's why they're called T cells, okay? T cells mature in the thymus, B cells mature in the bone marrow, okay? All of these cells, once they are mature, they get let loose in the body, okay? They are called at this point naive. They have receptors. They may have receptors for a particular antigen, but they have not actually seen it yet. They haven't seen it yet. They haven't been proven, okay? So they get released into the body. They're probably flowing around in the lymph. They may be hanging out on a lymph node when they first encounter the antigen that matches their receptor. When they find that antigen, encounter it, and it binds to their receptor, then you have activation, okay? So once a lymphocyte, B or T, is activated by receiving its antigen, it is now considered to be like fully mature. It's no longer naive. Now it's competent. And now it will basically reproduce. So now it's going to propagate and make more of itself because, hey, we have encountered this antigen. I am the cell that's whose job it is to recognize this antigen and launch an assault against it. So I will proliferate and produce many, many more of myself to outnumber this antigen. Okay, that's generally speaking, okay? These lymphocytes, before they can be allowed out into the body, APCs aren't the only way that they are, that we protect our body from these guys, okay? These guys need to go through a very rigorous training program. Remember, we have like famous finishing school, right? So that finishing school, this education program that the T lymphocytes specifically need to go through is referred to as positive and negative selection, okay? Now, when I say that it's learning, I don't mean that each individual lymphocyte actually learns a thing. Instead, you have a ton of lymphocytes and they're either good or they're bad. They either work or they're broken. And the broken ones are removed. They are programmed to die, so apoptosis. And the ones that are good, that are working good and don't attack self, those ones are allowed to go on and reproduce. So it's literally like natural selection of lymphocytes inside of your immune system. It's really trippy. And believe it or not, a very, very small percentage of lymphocytes survive this selection process. So it is very, very selective, because this is extremely important. Your lymphocytes need to be able to, one, recognize self. They have to be able to recognize the MHCs. They have to be able to recognize the major histocompatibility complex. If they do not recognize it, failure to recognize results in apoptosis. They pass that test, they get tested again. They also must not recognize self-antigens, okay? So basically, if they recognize self-antigens, as in if they fit with it and want to attack it, as in if self-antigens fit their receptor in the same way that a non-self antigen would, and it can't differentiate between it, apoptosis. You have to be able to recognize self, MHC, but you have to also not be able to recognize the antigens that our body produces. So if you can pass both of these tests, then you survive. If you fail one or the other, then apoptosis. More on this on page 794 of your text. Okay, you guys, make sure you read that. Okay, so here's a little compare and contrast between your B cells and your T cells. Your B lymphocytes, humoral. Your humoral immunity. T lymphocytes, cellular immunity. Do they secrete antibodies? B lymphocytes secrete antibodies. T lymphocytes do not. Okay. The primary targets for B lymphocytes, consider that they are humoral, so they're outside of cells. They are going to be targeting extracellular pathogens. Makes sense, right? So this is stuff that's floating around in the extracellular fluid outside of cells, as opposed to T lymphocytes, which are going to attack intracellular pathogens, inside of cells. Okay, so cells that are already infected, right? Or stuff that's inside of cells that's about to infect them, okay? Or cancer cells. Okay, so cells that are damaged, broken, that type of thing. They both originate in the red bone marrow, but the B lymphocytes stay in the red bone marrow to mature, while the T lymphocytes mature in the thymus. T, right? Effector cells, B lymphocytes work with plasma cells. T lymphocytes become cytotoxic helper or regulatory T cells. More on that in a little bit. And they both are able to form memory cells. Okay, so both B lymphocytes and T lymphocytes are able to form a memory of whatever antigen they're programmed to recognize in the way that they are programmed to recognize it, and in the region that they live in, right? They are both able to do that, okay? To form a memory of that antigen. Okay, APCs, a little bit more detail, okay? We talked about the differences between B and T lymphocytes. Let's talk about APCs. So this gorgeous thing right here is a dendritic cell, okay? Dendritic cells are a type of APC, okay? Type of, okay? APCs, they engulf antigens. So they phagocytize antigens or non-self stuff, and then they present chunks of their victims on their surface to T cells, okay? Which is kind of metal if you think about it. So they have little chunks of their victims on their exterior plasma membrane, which they then take to a T cell and say, look at this. Dendritic cells are ones that are found at the frontiers of the body. So basically like near the surface of the skin or near your orifices, places that where stuff can get in. So they're basically like front lines, okay? So they are also sentinels, which means that they sound the alarm first. They're the ones that are keeping a lookout, and they they alarm first. They notify the immune system, right? Okay. Macrophages can be APCs, and B lymphocytes can also be APCs, okay? So your macrophages, or your so these dendritic cells, all APCs, they are going to bring that antigen, or the chunks of the antigen, to the T cell, to a naive T cell, to show it its first antigen. Okay, so if the T cell encounters it on its own, it won't recognize it. It has to be brought to it by an APC. And that is like the final test of maturation, which is like so like, I don't know, like Game of Thrones or something. It's really kind of epic how that happens. So once T cells receive that antigen from an APC, they have to get it from an APC in order to recognize it. And then that's how they become competent, basically. That's how they go from being naive to being competent. Okay, so macrophages can also activate naive T cells. They also maintain T cell activation, and B lymphocytes also can present antigens to helper T cells, these guys, which aids in their own, as in the B lymphocyte activation. Gets a little complicated there. I'm trying to keep it as simple as possible. So basically you have B lymphocytes that are humoral immunity. You have T lymphocytes, which are cellular immunity. B lymphocytes produce antibodies. T lymphocytes do not produce antibodies. T lymphocytes are naive until they are activated by having an antigen presented to them by an antigen-presenting cell, which can be a dendritic cell, or a macrophage, or a B cell. Okay. All right, let's talk more about B cells now. Okay, so these are the guys that produce the antibodies. They're born in the red bone marrow. They mature in the red bone marrow. Humoral immunity, okay? Activation of B cells. So your B cells, so they have antibodies, right? So they've got these proteins embedded in their plasma membranes. That is what gives them their specificity, okay? When they encounter their specific antigen that matches with the antibodies on their surface, that activates them the same way that it does with a T cell, except it doesn't need to be presented by an APC. Then it will reproduce. It'll go through 2 rounds of division, right? So basically, these are somatic cells, so they will produce clones of themselves. The term clone is a little bit more important here because while these are the same type of cell, they're important individuals because they each have different antibodies for a different antigen, right? So this guy is the one that these antigens binded to because it had the correct antibodies for this particular antigen. So it is the only one that will start to reproduce. So it produces, it divides, forms 2 clones of itself, specifically with its antibodies. And then it divides again, which leads to 4 cells, right? 3 of those cells are going to become B cells that produce more antibodies. And 1 of them is going to be a memory B cell. So this guy is going to be in charge of looking out for this antigen again. And when it does, it's going to reproduce a ton more because it's going to recognize it faster and it's going to provide a stronger response, okay? So what you're looking at here, if you see a picture of a little cell that's got these antibodies on it, that's going to be a B cell. That's going to be a B lymphocyte because they produce antibodies. The antibodies can be embedded in the plasma membrane of the B cell, or they can be floating around, believe it or not. Okay, so antibodies they can be released into the bloodstream, and they often are. These guys' job really is to pump them out to be free through all of the interstitial fluid in the bloodstream. They are, like I said earlier, the little markers for an antigen. So an antigen binding to a B lymphocyte will look a little bit like this. But if these little antibodies by themselves find an antigen, they will still bind to it and they will mark it for death, right? They're going to mark it for destruction. They are a tag. They act as a tag for other immune cells like your T lymphocytes, your natural killer cells, macrophages, that this guy is a bad guy and he's marked for death, okay? So that's two different ways. It's like, it's a little bit weird, but I hope it makes sense that if the antigen finds a full-on B cell with the right antibodies, then that B cell will activate these antigens to the whole thing. So sorry, this guy will activate. We're going to produce a lot of antibodies. The antibodies will mark whatever other antigens are out there of this type for other immune cells to come in and destroy it and do the destroying part, okay? 2 rounds of division, 3 of them become antibody producing plasma cells or effector B cells. So that's what the plasma cell is, okay? If I didn't mention that before. And 1 of them will become a memory B cell, whose job it is to look out for this antigen again in the future and launch an even stronger response at that time. Okay, here is all this information is the same. Don't be alarmed. This is just showing you how the response is so much different between the first exposure and the second exposure. Okay, so the primary immune response occurs, it takes a little bit of time, a little tiny bit of delay, okay? This is time in days, so this is like, I don't know, what would you say, 1.5, maybe 2 days right here, between here and here, right? Takes a little bit of a delay. You have a response, which is, you know, good, but not huge, and then it tapers off. The second exposure, that response time has now been cut down to like less than a day, and the response is huge, relatively speaking. That secondary immune response is faster and larger, okay? Meanwhile, a new different antigen will start out with the primary response again, right? Okay, so summarizing humoral, the humoral part of the adaptive immune system, okay? So extracellular. You have active and passive. Your humoral immunity is active when you are infected with a virus or a bacteria or some other pathogen. You have contact naturally just from the environment. Then this whole process happens where the B lymphocytes produce antibodies, the T lymphocytes become mature and competent to recognize antigens and destroy them directly or indirectly. You can also artificially acquire an active humoral immunity by having a vaccine. You can have a dead or almost nearly dead, highly damaged version of the virus, usually a virus, injected into your interstitial fluid. So most of the time these are intermuscular injections, right? Vaccines are. So that goes into the muscle, probably doesn't go into a muscle cell. It's going to go into the fluid surrounding the muscle fibers. And that is going to come into contact with these B lymphocytes. Okay. And you're going to get that primary response. Which means that the next time, once you've been vaccinated, means that you didn't even have to have this primary response. You didn't have to get it. You didn't have to go through the entire thing because the version in the vaccine of this virus was damaged or dead, right? That would be much different than fighting a live virus. So having a damaged or a dead virus, you still get the antigen, but it's no contest. You beat it really fast. And then the second time you have the antibodies and you wipe it out and you don't even know that you got it, right? That's the idea. That's the idea behind vaccines. You can have dead or attenuated pathogens, or, and this is where I'm going to bring up the COVID vaccine, you can now have messenger RNA or mRNA, right? So the way that the COVID vaccine works is that instead of giving you dead or damaged COVID virus, which it wouldn't be obviously as dangerous as live virus, but even cooler would be to inject instructions for just these antigen proteins on the surface of the virus, which is what your immune cells, what your B lymphocytes actually recognize as non-self. It's those proteins on the surface of the pathogen, okay? Those are what link to the receptors. So instead of putting the actual virus in there with the external proteins, why not just put in that messenger RNA, which once it goes into the cells will find a ribosome, right? You've skipped the transcription because we didn't actually have this in the DNA. It never even goes into the nucleus. This messenger RNA just goes into the cell. It doesn't touch your DNA. It doesn't insert itself into your DNA. It does what every other messenger RNA that your nuclei produce does, which is find a ribosome and be translated into a protein. So your cells take this little messenger RNA just like it came from their own nucleus, which it didn't. It came from the vaccine. The ribosomes take it as it is, as written, and they follow the instructions. They build the protein. The protein by itself doesn't do anything. It's just a physical structure on the surface of the virus. So when that protein shows up, it gets made by the ribosome and it shows up inside of the cells and then getting passed outside of the cells. Being passed outside of the cells being the part that's going to be involving the B lymphocytes with humoral immunity. T lymphocytes would be involved inside of the cell. Either way, the protein is the antigen. So you don't even have to have the COVID virus inside of you. Instead, your lymphocytes recognize that protein as non-self and produce antibodies for that protein. Therefore, if you ever do get exposed to the actual virus, you have the antibodies for the proteins on the surface of that virus. You recognize it and you attack it immediately, and you're like far less likely to get seriously ill from it, which is awesome. So there's my little PSA. Go get your vaccine. It's cool. Do it. All right. Your passive humoral immunity can also be naturally or artificially acquired. You can have antibodies be passed down from mother to child, either through literally like being coded in them during like vaginal birth, or they are passed through the breast milk. This is a passive immunity. I kind of like to think of it as passing, because it's not very long lasting. So those antibodies being passed from mother to baby, they don't last for that long. They basically kind of give it a leg up until its thymus develops to the point where it's kind of developing its own lymphocytes, okay? So naturally, you're going to get some antibodies from your mom, but they're not gonna stick around that long because your body didn't make them itself. The ones that come from your thymus as you get a little bit bigger, those ones will stick around for the rest of your life, right? But mom is like saving your butt because you are coming out into this nasty world, right? And you want to like have as much advantage as you can get. You can technically also inject exogenous antibodies and get a passing humoral immunity to something, but it's really not the same and doesn't last as long as active humoral immunity. Okay, so getting the antibodies secondhand is no substitute for making the antibodies yourself in your own body. Okay. Because they won't be made by your B cells at that point. Your B cells still need to be trained to make that antibody, right, by themselves. So just having the antibody means that that antibody is going to eventually break down and there won't be anything to make it. until your B cells learn to recognize that antigen on their own. Okay, antibodies. We've been talking about them so much. Let's take a closer look, shall we? Your antibodies all have this kind of Y or T-shaped structures. They are proteins, right? So they're basically, they act as markers, they act as tags, and they have receptors on them to recognize a specific antigen, okay? So each one of these recognizes a specific antigen. They are actually 4 polypeptide chains linked up. So remember when we talked about like tertiary and quaternary structure of proteins? This is quaternary structure. So this is multiple peptides linking together to form a larger polypeptide, okay? So you've got this Y or T structure as a result. It's like semi-symmetrical, it's pretty cool. You've got 2 arms and a stem, okay? At the end of the 2 arms, you have an antigen binding site. So there's 2 antigen binding sites for each antibody. Okay, and again, these are produced by your B lymphocytes, right? By your B cells. Okay. All right, so your antibodies, so different ways that your antibodies can work, okay? So you have an antigen, bad guy. You have an antibody, which has been made to recognize a particular antigen, whether or not it's seen it yet, you know, who knows. But, oh, I forgot to mention, you have, it's not part of your innate immune system, it's still your adaptive immune system, but the reason why you don't immediately die when you get your first infection when you're a baby is because you do have a little bit of adaptive immunity built in. It's literally these antibodies, the proteins that make them, there's only a certain number of combinations possible when there are like billions of possible pathogens out there, right? So you have different combinations of antibodies like built in, antibodies for your B lymphocytes and receptors for your T lymphocytes built in according to what you evolutionarily will probably be exposed to in your lifetime. So you have a little bit built in based on your evolutionary history, which is really, really crazy. So you have antibodies for stuff that you may or may not ever encounter. A lot of your B lymphocytes and antibodies never encounter their antigen, so they never get used, which is fine. But for the antigens that we were not evolutionarily expecting to encounter, this is when we like make new antibodies and make memory cells for them, right? But we have a little bit built in already, which is really cool. All right, sorry. That's just fun facts. Really cool stuff. All right, so your antigen meets your antibody. You can have neutralization. The antibodies can surround the antigen and bind to it. A lot of antibodies binding to the antigen all over it literally like impedes its movement. It just like makes it slow down, it makes it bigger, it's bulky now, and it cannot get through a plasma membrane of a body cell, okay? So you are basically like creating a box or a shield around it so that it's unable to invade a body cell. Agglutination, you can have several antibodies kind of working together to hold a bunch of antigens together into a clump. Agglutination is what physicians or lab technicians look for if they are testing a blood type before a transfusion. So if you're getting a blood transfusion, of course, you want to know that you are getting the same blood type, right? Or else you're going to have major problems. The major problems is literally this will happen, okay? So you will have antibodies that will cause agglutination, basically cause that new transfused blood to clump. And if that happens in your body, then you're in really big trouble if you don't catch it in time. But you test blood destined for transfusion for agglutination before you actually use it for transfusion to make sure. And you look for this clotting, okay? And that's caused by antibodies. and precipitation. So you can have your antibodies making like a lattice with the antigens. And this, again, just makes like a really big target for other immune cells. So if you've got like this great big blob, like a big old clot, like in the case of agglutination or precipitation, you've got like a larger physical structure, it's a lot easier for other immune cells to come and find it and take care of it, okay? So these three things are all going to, they're gonna keep antigens from invading other body cells, and they're also gonna create a large target for phagocytes, right? For your macrophages, and your neutrophils. So creating large targets for phagocytosis. You can also have these antibodies binding to antigens can also call those complement proteins, okay? So the antibodies themselves, they create a marker on the cell. Then if a natural killer cell finds it, or if a T lymphocyte finds it, it sees these markers and it's like, you are scheduled for death, my friend. Then they will either take care of it themselves or they will call in complement proteins. And those complement proteins, again, will embed themselves in the plasma membrane of the pathogen and form a hole and cause it to lyse, okay? So this also is going to, this marking, this triggering, this marking of the pathogen is going to also can also call over phagocytes, and it can also help to trigger inflammation. So if a natural killer or a T lymphocyte finds this, a marked cell, they may also just trigger a further immune response. They might trigger inflammation and call more immune cells over. That leads to cell lysis. Okay. So your antibodies work in a couple of really cool ways, right? They can like do a citizen's arrest of an antigen and hold it and keep it from invading anything else. And they also mark it for death, either by macrophages or other immune cells, natural killer cells, T lymphocytes, okay? Competent T lymphocytes, remember, okay. Ones that have already seen it and can recognize it directly. Okay. Cellular, so we've talked about humoral immunity. Okay, so all of this stuff is taking place outside of the cells. Let's talk about T lymphocytes and how they act in cellular immunity, intracellular immunity inside of the cells, okay? So you've got two different major types of T cells. This is literally based on what's called the cell differentiation glycoprotein. So cell differentiation 4 means that it has glycoprotein 4. And cell differentiated glycoprotein 8 is what that type of T cell has. Okay, so it's literally a glycoprotein on their surface. It's an identifying protein. So you've got your little immature lymphocyte in the red bone marrow. They are going to go to the thymus, which means that they're going to become T lymphocytes, right? You can have some that are going to turn into CD4s and some that are going to turn into CD8s. They basically have they have their own receptors, right? They will be introduced by a APC, right? An antigen-presenting cell, this little cute little dendritic cell guy. They're going to be introduced to their antigen that they were made for, right? And when they are introduced to it, they become competent. They go from being naive to now being competent. They will reproduce and they'll create a memory cell for every 3, or I guess for every 4, or every, one complement of every, one memory cell out of every 5 progeny. The rest of the progeny will become your little T lymphocytes going around looking for that antigen forever and ever. And then CD8 just has a slightly different looking receptor, okay? Just a different glycoprotein going on there. There really isn't much of a difference there, okay? But this, I didn't want this figure. I like this figure, but I didn't want the fact that there's 2 different pathways here to freak you out. Consider them to be, generally speaking, kind of the same pathway, except that they have different glycoproteins, complementary or cell differentiated glycoproteins. Okay. So don't worry about specifically the CD4s become helpers and CD8s become cytotoxic. Some T lymphocytes become helper T cells and some T lymphocytes become cytotoxic T cells, okay? All right. A little bit more on this. I'm actually not even worried about this one. So please disregard this slide. All right. So this is a cytotoxic T cell. Let's go over a little bit of how a T cell actually goes about its cellular immunity functions. Okay, so this cytotoxic T cell is going to find a target cell. So it's going to encounter its antigen out in the wild after one of these little guys, basically. After he's been made competent, we've reproduced, all these little guys are considered to be competent helper T cells. I guess I would be over here. They competent cytotoxic T cells, excuse me. Okay, so these guys are now released out into the body. Maybe they're going to be looking for infected cells. Okay, their job is to do intracellular immunity. So the B lymphocytes are busy taking care of the stuff happening in the extracellular fluid outside of cells. These guys, your little cytotoxic T cells and your T helper cells indirectly, are going to be looking for cells that have been marked either by antibodies or by a lack of MHCs, right? That major histocompatibility complex. Either way, it binds to that target cell, and then it's actually going to like inject what are called perforins, which actually like perforate the membrane, okay? So they go in and they form granules and they cause apoptosis of the target cell, okay? So you're going to see exocytosis from the T cell over here, exocytosis of perforins, which are just like toxic proteins, basically. There's going to be exocytosing that at the surface of the target cell over here, okay? Those perforins are going to go in and basically trigger apoptosis. And this target cell, again, is marked by either antibodies or by a lack of MHC, or some cells like cancer cells, like they will mark themselves in a way that certain cytotoxic T cells can recognize to basically say like, please end it. Please end it. Something's wrong. Okay. All right. Cells of the immune response, both humoral and cellular, okay? B cells, humoral, right? Plasma cells formed by your B cells produce antibodies. They produce huge numbers of antibodies, okay? Your plasma cells. The B cells are lymphocytes that form plasma cells and memory cells that make antibodies. Very, very important. Hence why I keep repeating myself ad nauseum. I'm sorry. All right, your T cells. You've got a few different types. Don't worry too much about the specific differences between the different types. But they do not have antibodies. No antibodies from T cells, okay? These guys, once they go from being naive to being mature, which means that they receive their antigen from an APC. Does that say that in there? Cytotoxic plasma cells activates macrophages. They need to be activated by an APC, okay? And your memory cells, again, you can have memory cells come from the progeny of B cells or from T cells. Either way, their purpose is to recognize and react to that same antigen the second time around faster and more strongly, okay, in whatever way that they do, whether it's humoral, B lymphocytes, or intracellular T lymphocytes. and APCs, right? So these guys. Dendritic cells are the most common example of an APC. They are going to have MHC, that major histocompatibility complex, for the T cells to recognize. And they also are going to kill antigens, attach them to themselves, and present it to the T cells so the T cells can get their first taste of that antigen from an APC, and that keeps T cells from going crazy, okay, and starting to like kill everything. They have to be activated appropriately by an APC before they are allowed, before they're set loose on the rest of your body cells, because they're going to be a huge autoimmune problem, right? Turn on my light here. We lost the sun. Okay. Molecules. Okay, so here we have cells of the immune response. Here are some molecules of the adaptive immune response. They include antigens, which again are, they're not technically the actual pathogen itself, but it's proteins on the surface of the pathogen. Okay, so that's why it's a molecule. So it's going to be a substance capable of provoking an immune response, which can be really any type of macromolecule and other weird chemical things. But in the case of like viruses or bacteria, it's usually like a protein associated with the plasma membrane, the outer plasma membrane of that pathogen, okay? That is not normally present in the body, hence why it's non-self, right? Okay, antibodies, also just a molecule, right? These are polypeptides exhibiting quaternary structure, right? They are produced by B cells, right? Or by the plasma cells that the B cell produces when it first encounters that antigen. They're released in the body fluids, they attach to antigens, they cause complement activation, neutralization, precipitation, agglutination, right? Which is all that stuff from over here. So all the stuff that antibodies do is also described here. We also have perforins and granzymes. These guys are the guys that the T lymphocytes use. The T cytotoxic specifically, if you want to know, but T cells will suffice. They use that to target the cell's membrane, which is going to cause apoptosis, okay? Complement proteins, blood-borne proteins. They are activated by various immune cells, and they basically also, they're gonna form a hole in the wall of the targeted pathogen, which is gonna cause water to flow in and for them to lyse and cause it to burst, okay? And cytokines, not super worried about it. The ones that I'm underlying here are gonna be the really important ones. Antigens, antibodies, complements, maybe perforins. But cytokines, well, I mean, maybe kind of like, consider maybe histamine. These are basically going to be the chemical signals that initiate inflammatory response and further immune response, activating the immune system. I want to say that they activate inflammation. That's going to make more sense, I think, to my brain anyway, hopefully to yours. Okay, so they activated immune response. Oh, wow. I swear. I've got bad lag happening over here. Bear with me. Slash inflammation. Okay. All right. So sorry, it's all important. Okay. Okay, so I want to reiterate this again. We've already seen this at the beginning of our adaptive immune system, okay? Adaptive immune system, I'm sorry, your immune system in general. We talked about this first. Seems like so long ago, right? You have your innate defenses, your surface barriers, skin and mucous membranes, your internal innate defenses, your phagocytes, natural killers, inflammation, fever, and antimicrobial proteins. These things that are more generalized and will basically attack anything that's recognized as non-self. And then your adaptive defenses, which they like proliferate hugely. It's a very, very strong response. It occurs outside of the cells, which is called humoral immunity and involves your B lymphocytes, or inside of the cells, which is called cellular immunity and involves your T lymphocytes. And it involves memory, okay? It involves antibodies and memory, right? Okay. The innate defenses are very general, and everybody has them built in. They're innate. Okay. Adaptive is also maybe called acquired. So don't let that fool you. This is a beautiful illustration found on pages 808 and 809 in your text. So this basically shows kind of the entire immune system going on here. So you've got your first line of defense. So in the case of like you got a cold or something, so you've got the flu, influenza virus. All right, so it got past you. What you gonna do, right? You breathed it in somewhere. You walked through somebody's like cough cloud or sneeze cloud or something gross, and it made it into the airways. The mucous membrane of the airways is going to help to try to catch it before it gets any further and sweep it away, right? So the epithelial cells of your airways are ciliated, right? You also have goblet cells, which are producing mucus, okay, in this mucous membrane of your airway. That mucus is going to catch the virus and sweep it out. Further, you will have an inflammatory response. If these cells are encountered by a phagocyte, like this macrophage, then it's going to release chemical signals, cytokines, and that is going to cause your blood vessels to dilate, for your capillaries to become more permeable. Then you're going to have more immune cells arriving that are able to squeeze through the walls of the capillaries to get to the virus, to get to the problem, right? And meanwhile, some of the viruses are taken by complement proteins, right? So they're marked and taken by complement proteins. They're lysed that way. Other immune system cells, like your T lymphocytes right here, they are going, oh, never mind, this is natural killer cells, excuse me. So your natural killer cells can destroy them directly as well. That is part of your second line of defense, right? So the first line of defense is all this inflammation response and the physical barrier of the mucous membrane. Second line of defense is your internal defenses, which is like your natural killers, more general, killing things that are not self. And then finally, your third line of defense is going to be involved when your dendritic cells, which should be the sentinels, right? They should be right here on this front line. They also are going to find virus. They will destroy the virus, phagocytize the virus, and then they will actually display chunks of the virus on their surface. And then they will make their way to a lymph node to find a T cell, a naive T cell. And then we go into this right here. The naive T cell, it receives the antigen from the APC, which is the dendritic cell. That antigen-presenting cell is going to cause that T lymphocyte to activate, and it will divide and form 3 effector cells for every memory cell. The effector cells are the ones that basically get set loose in the body and are going to basically go around destroying infected body cells. The B lymphocytes, meanwhile, will be going around, and if they recognize and are activated by the antigen, they will produce antibodies, which will go out and find the antigen themselves, and they can like interfere with them by neutralizing them. They can cause agglutination, or they can call over complement proteins to lyse the cell that way as well, right? Meanwhile, your little T cells that have been let loose, here's your B cells forming your antibodies, forming plasma cells, which produce antibodies. Here's your T cells. They go out and they are going to use those perforins to kill infected body cells, right? Because their job is to destroy the intracellular invaders. So once it's intracellular, that means that that cell is infected. So it kills infected cells. The B lymphocytes, therefore, produce antibodies that go after the antigen itself out when they're outside of the cells and haven't invaded into an actual into a cell yet, right? So hopefully this gives you an idea of one, the many layers of the immune system, from your first line of defense, your physical exterior barriers, your second line of defense, those innate cellular general barriers, and then your third line of defense, which is your adaptive immune system. T lymphocytes, B lymphocytes, T cells going after the infected body cells, and B cells going after the actual pathogen when it's floating around in between cells. So you've really got all the bases covered here by your body's immune system, and it's really, really cool how it works. I hope that you think it is cool, because I think it's really pretty cool. A couple of things that can go wrong. So let's talk about some stuff that can go wrong. Immunodeficiency, right? So if something causes the production or the function of immune cells to be impaired, then you are unable to launch an immune response against a pathogen. There are congenital immunodeficiency problems. SCID is severe combined immunodeficiency. There are multiple possible syndromes that fall under this category. And they are all congenital, which means that people are born with them. So having a congenital immunodeficiency, it's basically like the boy in the bubble. If you have a compromised immune system that way, it's really hard to lead a normal life, because you need to protect yourself constantly from everything. Because if you were to contract a pathogen of any sort, you have no way to fight it. You can also have acquired immunodeficiency disorders. Hodgkin's lymphoma, a type of cancer of the lymphatic system, can destroy your lymph nodes and cause problems with their function, therefore the immune cells inside of them. Immunosuppressive drugs. So if you are battling cancer, or if you have a transplant or a transfusion, you are going to have to take drugs that are going to suppress your immune system on purpose, so that your body doesn't hurt itself, basically, right? You take immunosuppressing drugs if you have a transplant or a blood transfusion to keep it, or to try and like dampen the response of your immune cells to what might be recognized as non-self. HIV/AIDS. So HIV is the human immunodeficiency virus. That virus causes the condition known as acquired immune deficiency syndrome, which is AIDS, acquired immune deficiency syndrome. So if you have HIV, you can potentially fight it and win. But if HIV turns into what they call full-blown AIDS, then at that point, your immune system has been compromised irreversibly, and there is no cure. There's no cure for any of these, for really any immunodeficiency disorder, because there's no way to activate an immune system that has been compromised that we have found yet, unfortunately. So there isn't really a good prognosis at this time for people with this kind of acquired immune deficiency syndrome, whether it comes from AIDS or whether it comes from HIV, the human immunodeficiency virus, or elsewhere. It's a matter of preventing infection all day, every day, for as long as you can. The opposite problem to having an insufficient immune system is having an overactive immune system. And we call these autoimmune diseases, okay? So this is when the immune system is unable to distinguish friend from foe. So it's not like the immune system cells are not working, they're not functioning at all, they're functioning too much. And instead of being able to differentiate between self and non-self, they just start attacking everything. So those MHCs, the major histocompatibility complex, starts to not work in keeping immune cells from destroying healthy body tissue. You can prevent it, or we do prevent it in the vast majority of cases. People that don't have autoimmune diseases, it's prevented by that training that we talked about, that negative selection. So if a T lymphocyte in training fails the test, fails that training test, remember it gets apoptosis. If it fails to recognize MHCs, then it's going to be eliminated, ideally. The other is the activation of T cells requires that APC. So T cells can't be activated until they get the antigen presented to them by an antigen-presenting cell, right? Those dendritic cells. That is a safety check to keep these T lymphocytes from going crazy. If something goes wrong, and most of the time we don't understand really what it is, which of these is going wrong here, you can end up with a full-blown autoimmune disease. Autoimmune diseases affect 5% of the US population. 2/3 of those affected are women. Weird, right? Rheumatoid arthritis, autoimmune disease, attacks the joints specifically. MS, multiple sclerosis, autoimmune disease, attacks the myelin around the central nervous system cells, right? So remember the myelin sheath is that white fatty substance that surrounds the axons of neurons and allows for the signal to pass through, which is why MS sufferers have a harder time with voluntary muscle control, among other muscle control. Type 1 diabetes, insulin-dependent diabetes. This is actually sometimes congenital, but it has nothing to do with lifestyle, diet, exercise, anything like that. Instead, your body starts to attack your insulin producing and your insulin or your insulin receiving cells. So you end up having to be treated for that. Otherwise your body, so your body will not produce insulin. So your insulin producing cells are destroyed. They're targeted and destroyed for some reason. So you end up having to have insulin injections. The treatment for autoimmune diseases is really just to block immune molecules. You can use immunosuppressive drugs, but like we were saying, immunosuppressive drugs, they work on the entire immune system. So it's really a problem to be immune compromised. It's potentially a worse problem to be immune compromised than it is to have an autoimmune disease. So it's kind of it's a balancing act, right? But the only way to do it is there are some medications, treatment plans out there for blocking specific immune molecules, but not others. And some other like experimental treatments that were mentioned on page 812, which is pretty interesting if you want to check that out. But I know we're really running short on time here as I stare at my naked wrist because I forgot to put my watch back on. All right, lastly, and one that I think we're all very familiar with, are hypersensitivities, also known as allergies. We hate them, right? Hypersensitivities damage tissue or tissue damage caused by the immune system as it's trying to fight against a perceived threat that's not actually dangerous, like pollen or pet dander, peanuts, nuts, shellfish, right? Anything else that you can be allergic to. It's been perceived as an antigen. Whether or not it's actually dangerous to the body, oftentimes it's not, and it's really just a pain in the butt. It usually doesn't do any more harm than just being very, very annoying. You have two different kind of subgroups of this. You have immediate hypersensitivities, which are basically like your normal kind of like respiratory allergies, and foodborne or food allergies. Anaphylactic shock, which is basically a systemic response to usually a food allergy, maybe sometimes like a bee sting or something. It's a systemic reaction to one of these not actually dangerous, not actually like lethal threats that can actually cause death. So part of that reaction is to, to vasodilate throughout the entire body. It can actually cause you to go into shock, constricting of the airways, obviously dangerous, not good. The immediate treatment there, of course, is going to be your EpiPen, right? That's epinephrine. Epinephrine to the rescue, right? And then delayed hypersensitivities are things that are more like contact dermatitis. So like I am allergic to nickel in cheap jewelry. I can wear it for a little while, sometimes like a day or two, maybe just 12 hours before I develop a rash. So you have that you have that delay between the exposure and the, the resulting tissue damage, if you will, the reaction. All right, so that's what allergies are. They're just hypersensitivities to things that really shouldn't be lethal or dangerous in any way, but your immune system has somehow recognized that molecule as an antigen and has formed a memory of it. So the first time that you get exposed to it, just like with any other antigen, it may not be, for an allergy, it may not seem like a big deal. You may not even notice it at all. But the second time around and thereafter, once you have antibodies for that thing, whatever it is, the response is going to be fast in the case of an immediate hypersensitivity and very, very strong. Okay. All right. And that's it. So let me real quick show you what we're going to be working with in lab this Thursday. I've made you an in-depth description this time because I know you guys like a little bit more direction. So I'm trying to do that for you. We're going to continue with our rabbits, but we are going to be looking at the endocrine system, which should be pretty cool. So we won't be able to see the stuff in the brain. We're not going to be cracking any skulls on Thursday, so please don't. But you will be able to open the body cavity and hopefully find the thyroid. The parathyroids will be very difficult to differentiate unless you look very closely, but you are, of course, encouraged to look. The thymus, probably not going to be able to find. It's going to be very small, associated. It's going to be near the heart. Your pancreas in the rabbit is also going to be very difficult to find. It actually kind of looks like the rest of the, or it's just kind of like a tissue outcropping of the small intestine, of this part of the small intestine. So you have to be careful looking for the pancreas. The adrenal glands are located not directly on top of the kidneys, but actually a little bit medial to the kidneys. So keep an eye out for that. And whether you have if you have a female rabbit, you're going to be looking for ovaries, and in a male rabbit, testes, of course, right? So that is going to be all of your endocrine glands you're going to be looking for in your rabbit. You really need to be able to identify these by their location. That's really what's important here. Okay, these main ones. The pituitary, yes, you need to know where that is too, but even though we're not looking at it in the dissection, but the thyroid, parathyroid, pancreas, adrenals, and ovaries or testes. Those are all, I should say, and or testes, because it is possible to have both, but the odds are you won't be getting one of those, sorry. We also have the slides from last time. Those are still out, so make sure that you take a close look at those with your microscope. And I have some images of what you can possibly look for, as well as some resources to look for in the slides, right? as well as some more resources in case you don't have a slide that has a really nice example of these structures. But for each of these, see if you can't differentiate or find each of these different cell types in here. It's just going to be really good practice for you, okay? What you specifically need to know will be on your study guide, which I'm going to post by the end of tonight, which is Monday. So here's a PowerPoint of these images and more. Here's a link to Lee's virtual histology lab where you can find those beautiful endocrine slides that you can zoom into and find different structures in. This is a worksheet that some of the other labs are working with. If that interests you, it's answering questions based on the lab, and these images are from that. So if you wanted to have a little bit more direction, you are more than free to print this out and work from this worksheet. That's totally cool too. All right, you guys, so that's Thursday. And have a great week until then. All right, you guys, I'll see you Thursday at 1. Bye.